Hello my geniuses, welcome to another lecture series today. It's your great science tutor LAWRENZO, and we're talking about the auditory pathway. That is, how do your brain understand what is said? So the sense of hearing and understanding, that's what we are talking about today. So we have done the olfactory pathway, we have done the visual pathway, we have done the gustatory pathway. So now we are in the auditory pathway. These are just special senses. Now under the special senses physiology, your special senses pathway. So if you are new to my channel, make sure to subscribe and turn on your post notification button so that anytime I drop any video, you'll be sure to be notified. And if you want me to make a video for you, drop it down in the comment section. I promise I'll make that video solely for you. So let's jump right into the video. So the first one we'll be talking about is the introduction to the auditory pathway. So how did you listen? How did you hear? So we'll be starting with the introduction to the auditory pathway. Same as the olfactory and other special senses pathway, it is also a neural pathway. We know all of them are all neural pathway that carries what movement and transmits sound. Here it coordinates movement and transmits sound from the inner ear to the what cerebral cortex. So it carries the sense of what hearing, or carry movement first, which means you have to coordinate your head and present it in a way where that sound can enter it. So movement and what transmission of sound through the inner ear. You get it now? So from the inner ear to the what cerebral cortex. So we have the auditory cortex in the cerebrum. So it helps us to interpret what we have heard, whether it is something we can understand or something we cannot understand. So again, it's a neural pathway which involve movement. Are you getting me? And transmitting of sound from the what inner ear to the what the auditory cortex in the cerebrum. Are you getting me? Where they are interpreted and sound or occur. So where they are what interpreted. So the next one, what is the stimulus? Remember, after your introduction we have the stimulus. So what is the stimulus of sound? The stimulus of sound is anything. Somebody can be talking to you, a moving car, a cock crowing, a dog barking, anything that you can interpret in your brain, anything you can understand in your brain. So that's what we want to talk about, sound. But let us use sound from a moving car. So how can we recognize that? Okay, a car is coming from our back and we have to leave the road so that it cannot hit us. So how can we understand that it is a car coming? So, and how can we differentiate a car from a baby crying and all these things? So that's what we want to talk about. First of all, what is our receptor? Don't forget, our receptors are usually dendrite of bipolar neurons. Don't forget, dendrite of bipolar neurons. But this receptor also is dendrite of bipolar neurons, but they are usually located around the hair cells. So the dendrite of bipolar neurons are around the word hair cells. So we have the inner hair cells and we have the outer hair cells. But for the purpose of this lecture, the inner hair cells are innervated more by sensory neurons than the outer hair cells. So the hair cells of the word spiral organ of what Corti. So the spiral organ of Corti is located inside the inner ear, and it is this sensory organ, or it's a sensory tissue that we use to what hear. So it is that part that is being stimulated that can start the nervous system pathway from that inner ear to the brain. So the hair cells of the spiral organ of what Corti. So now remember, in the spiral organ of Corti, they are dendrite of what bipolar what neurons. So when they are dendrite of bipolar neurons, they carry their impulses through the what axons, as we all know it. So if you stimulate the dendrite, which is the hair cells, it will what reorganize and resynchronize in the cell body. And when it organize and resynchronize the cell body, it's carried out through the what axon. Remember, action potential for sensory neurons are originated in the first node of Olivary. So when action potential is gotten, it now transmits through the word axon by sortatory conduction if they are myelinated by Schwann cells. Remember, sensory neurons are myelinated by Schwann cells, same as motor neurons, because they are peripheral neurons. But CNS neurons, those in that the tracts that come from the brain down to the spinal cord, are myelinated by oligodendrocytes. Don't forget. So now we are talking about the myelination. They are myelinated by Schwann cells. So when it is action potential is gotten from the first node of Olivary, they undergo what we call sortatory conduction. So when they undergo sortatory conduction, what happens? The axon snaps in the what we call the spiral ganglion. So that is the first stop it takes. Immediately enter to the dendrite which is in the hair cells. It resynchronizes in the cell body, moves to the axon, and snaps on the spiral ganglion. So that's the first stop. In the spiral ganglion, where is the spiral ganglion located? In the modulus of the cochlea. The modulus of the cochlea. That is where the spiral ganglion is located. So that is where it takes its first stop. Now, from that first stop, what happens is the fibers from that spiral ganglion now fuse together to form what we call the cochlear nerve. Don't forget. The first stop, which is the axon, snaps on the spiral ganglion located in the modulus of the cochlea. So when it is snapping there, afferent fibers from that spiral ganglion originated and fused together to form the cochlear nerve. So now we have the cochlear nerve. The cochlear nerve is a part of the vestibulocochlear nerve, which is the cranial nerve number 8. You get it? So the vestibulocochlear nerve. So the vestibular coordinates kinesthetic movement, balancing, you get it, vibration, equilibrium, while the cochlear nerve carries hearing, because in your ear you use your ear for two things, for balancing and for hearing. So when the cochlear nerve, when the fibers which fuse together to form the cochlear nerve, they passes through what we call the internal acoustic meatus. Now the internal acoustic meatus, they pass through the internal acoustic meatus together with the facial nerve. Remember, the facial nerve, the vestibulocochlear nerve, and the labyrinthine artery passes through the internal acoustic meatus to go towards the brain. So they leave the skull through the internal acoustic meatus to go to the brain. Now this is the first order neuron. Remember, bipolar neurons, which is the dendrite, is located in the hair cells, which is usually much important in the inner hair cells. So when they originate there, they are synchronized in the cell body, pass through the axon, and snaps on the spiral ganglion. Afferent fiber from the spiral ganglion leaves and fuse together to form the cochlear nerve. So the cochlear nerve is a branch, or is a part of the vestibulocochlear nerve, which is the cranial nerve number 8. So after they pass through the internal acoustic meatus together with the facial nerve to leave the skull. So when they leave the skull, now in the second order neuron, what happens now? The cochlear nerve snaps on what we call the ventral and dorsal cochlear nucleus. So the ventral and dorsal cochlear nucleus are located in the pons, in the lower level of the pons, because the pons anatomically can be divided in the upper level and in the lower level. So when you divide, or when you dissect in transverse section in the lower level of the pons, you can get the ventral and dorsal cochlear nucleus, which is at either side of the nucleus of the inferior cerebellar peduncle. So if you look at the lateral side, when you are looking in the inner structure of the pons, at the lateral side, you will see that the ventral and dorsal cochlear nucleus are at either side, which is front and back, ventral and dorsal part of the inferior cerebellar peduncles, the nucleus of the inferior cerebellar peduncles, don't forget. So when you see there, what happens now? Fibers from each side of the ventral and dorsal cochlear nucleus, they cosseate. What is the meaning of the cosseate? They cross each other. So ventral from the right side and ventral from the left side crosses each other. So when they crosses, what happens? They form what we call a trapezoid body. So a trapezoid body is a white matter fiber. It's a decosseated white matter fiber. So when they cross each other, the right and left cross each other, they form a trapezoid body. And when they form a trapezoid body, they take their second stop. Remember, the first stop is at the spiral ganglion. This next stop is after they decosseate, they snaps on the superior olivary nucleus. It snaps on the superior olivary nucleus. So the superior olivary nucleus, you can see the superior olivary nucleus also at the lower level of the pons. So when they snap there, what happens? Also, not only the contralateral fiber, not that the contralateral fiber snaps on the superior olivary nucleus. Its bilateral fiber from the ventral and dorsal nucleus can also snap laterally on the same side on the superior olivary nucleus. So we have a contralateral fiber from the other side of the ventral and dorsal cochlear nucleus, and also its bilateral fiber from the ventral and dorsal cochlear nucleus snapping on the superior olivary nucleus. You get it now? So remember, after the cochlear nerve leave through the internal acoustic meatus, they snap on the ventral and dorsal cochlear nucleus, which is at either side of the inferior cerebellar peduncles. So when they are there now, what happened? That is their second stop. Then what do? They decosseate, which means they cross to the other side. The right one cross to the left one cross to the right. So when that crossing they are taking, it forms what we call the trapezoid body. So when it forms the trapezoid body, it snaps on each other side on the superior olivary nucleus, but fibers from the ipsilateral same side of the of the ventral and dorsal cochlear nucleus also reaches the superior olivary nucleus of the same side. So they have the contralateral fiber from opposite side and ipsilateral fiber from same side. So when they are in the superior olivary nucleus, some fibers from the superior olivary nucleus of the same side carries it and snaps on the nucleus of lateral lemniscus. So in the nucleus of lateral lemniscus at the upper level of the pons, so that is where they snap. So when they snap in the nucleus of the lateral lemniscus, then remember, after they cross, they snap on the superior olivary nucleus. Now from the superior olivary nucleus of the same side, first of all contralateral side and same side, fibers from that place on the superior olivary nucleus of the same side carries it up to the nucleus of lateral lemniscus in the same pons. So when it carries it there, then lateral lemniscus fiber, there's what they call a lateral lemniscus fiber, because there are different lemniscus. You have the lateral, we have the medial, we have the spinal and the trigeminal lemniscus. But in this case, the lateral lemniscus fiber carries it from the nucleus of the lateral lemniscus to the inferior colliculus. So they carried it to the nucleus of the inferior colliculus, don't forget. And the nucleus of the inferior colliculus is located in the midbrain. They are located where? In the midbrain. So again, from the superior olivary nucleus of same side to the nucleus of lateral lemniscus in the upper level of the pons, to the nucleus of the inferior colliculus using the lateral lemniscus. But fibers from the superior olivary nucleus snaps it on the nucleus of the lateral lemniscus. Then lateral lemniscus white fiber carries it from the nucleus of lateral lemniscus to the inferior colliculus, the nucleus of the inferior colliculus. When you give a transverse section of the midbrain at the level of the inferior colliculus, you will see the nucleus of the inferior colliculus there. Now there's what we call the inferior brachium. The inferior brachium carries the impulse fibers from the inferior colliculus, which is the nucleus of the inferior colliculus, to the medial geniculate body of the thalamus. So remember, the thalamus is a relay station where all impulse fibers going to the cerebrum pass through first. So from the nucleus of the inferior colliculus, fibers carries it to the medial geniculate body, which ends up the second order neuron. You are seeing it on the screen. Now in your third order neuron, there's what we call the thalamocortical fibers. The thalamocortical fibers connect the thalamus and the cerebral cortex. So the thalamocortical fibers carries this information from the thalamus, which is the medial geniculate body, and down to the what we call the inferior radiation. So when they take it to the inferior radiation, they snaps on the primary auditory cortex, which is area 41 and 42. The primary auditory cortex is located in the temporal lobe. So when they snaps on the primary auditory cortex area 41 42, you tend to interpret what you just had. You tend to now understand that it's a car. But now I will teach you about the movements, how the brain coordinates later. But you have to know that you interpreted that, okay, this is a car coming from the sound. Primary auditory cortex area 41 and 42, and also the auditory association cortex, which is the Wernicke's area 22, still in the level of the temporal lobe, but at the side of what we call at the terminal end of the lateral sulcus. So the terminal end, when you are going outside into the lateral part of the brain, you sit there. So you have the Wernicke's area 22. This is how it is. So this is the third order neuron. You get it now? Now for the clinical correlation here, what are the clinical correlation? We have what we call the conduction deafness and we have the neural deafness. The conduction deafness is just a partial deafness, which means when you settle the issue or you treat yourself from what is blocking the ear, sound will pass through easily when you remove the barrier. So why neural deafness is that it's a complete deafness because the neural pathway, which we have discussed, one of them are damaged. Now in the conduction deafness, you can either have a damage or obstruction to the external or middle or internal ear, which affects sound traveling. So sound transmission can be impeded in either the external ear where there's wax or water or anything. In the middle ear, maybe anything can be there, maybe fluid or infection or anything. Or in the inner ear, something happens to the inner ear, it swell up or something happened, which prevents sound traveling, which means the ear is fine, it's just that the sound traveling is impeded. So what can be the causes of conduction deafness? You have blockage of the external ear by wax, which we have talking otitis media. But in the otitis media, is either the wax is the cause of breeding of bacteria which affect the ear, or you have what, you perforate the tympanic membrane, which is the eardrum, and when you perforate the tympanic membrane, sound cannot be stimulated on the tympanic membrane. It will just leave as air and you will not perceive any sound. And thickness of the tympanic membrane due to repeated infection. So when the tympanic membrane is infected continuously, it becomes thickened. Even when air strikes on the tympanic membrane, it will not stimulate, or it will not vibrate the malleus, the handle of the malleus on it, so nothing will happen. Also, fusion of the step on the ovary window. Remember, the steps on the ovary window, when sound travel to and a bit on the eardrum, it vibrates the handle of the malleus. So when you vibrate the handle of the malleus, then the handle of the malleus, the head of the malleus is attached to the incus, which also conducts the sound from malleus to the incus, then from incus to the stapes. Then the stapes move in and out of the ovary window, causing sound to travel to. So when there's fusion of the stapes on the ovary window, which means sound cannot travel to. So you get it now. So what about the neural deafness? Neural deafness is when there's a damage to the cochlear nerve or any part of the auditory pathway. So when there's damage, you cannot transmit sound. So it can be used when you are using drugs like streptomycin, which is amikacins. So when you are using streptomycins, you are using gentamicins. You get it now. So when you are using any of this drug, it will affect you very well. You get it now. And also it damages the hair cells. So also tumor pressing on the cochlear nucleus. It could be any part of the pathway. It could be in the nerve, it could be in the nucleus, or it could be in the brain. Anyone, you will not just hear. It's a complete deafness. So how can you assess hearing? How can you assess hearing? So it's simple. You can assess hearing through what we call the whisper test. Okay, now, so you can do it through the whisper test. In the whisper test, what happens? The patient hear you while you are whispering. So you just stay 60 centimeters away and you whisper. And when you whisper, if the patient can hear you, which means there's negative conduction deafness, so the patient is fine. So you have what we call the Rhines test. In the Rhines test, what happens? You put a tuning fork, place it on the mastoid, because you want to check for unilateral conduction deafness, not contralateral conduction deafness. You just want to check whether one ear is good and the other ear is not good. So unilateral deafness. So when you put it on one side of the ear, you beat the tuning fork and place it on the mastoid bone. You want to check for vibration, whether the patient can feel vibration. And later, when the vibration stops, then you hear the sound from the tuning fork. If the patient cannot feel it, which means he has unilateral deafness. Are you getting now? Then you now have what we call Weber's test. So Rhines test is for tuning fork on the mastoid, while Weber's test is beat the tuning fork and place it on the vertex of the skull. So when you place it on the vertex of the skull, you want to check for bilateral you want to check whether the bilateral ear is working. The two ears are working, or maybe there is conduction deafness in the two ear. Are you getting now? You are assessing for lateralization of sound and also the difference between conduction deafness and neural deafness. So the last one here is using an auditometry. An auditometry is a device which is used to sense the not even conduction deafness. You want to check whether the cerebral cortex can interpret sound and also whether the patient can really hear sound. So you want to force the sound down to the cochlea, not really for the conduction. You get it now? So I know the hearing of human can be up to greater than 30 decibels. So when you send it's just like using an auditometer. Auditometer is just an amplifier. So when you switch the auditometer and the patient puts just like the headsets kind of version on the ear, so when the patient puts it and you increase the frequency about 30 to 40 decibels, then it transmits to the ear. So it's used to check for complete deafness. You won't check for neural deafness, while others are used to check for maybe conduction deafness, but this auditometry is used to check for neural deafness, whether a patient can really hear or not. So hope you understand what I've given you today. You get it now? So we started with talking about the neural pathway, which is carrying sound impulses from the inner ear to the brain. Are you getting me now? So we say the stimulus can be car moving or sound kind of a baby. The receptors are in the hair cells. So when the hair cells, when the sound comes to the hair cells, they move just like your normal hair. They move, and they are dendrite of bipolar neurons. So when they pick up the sound, they move it, synchronize it in the cell body, the axons. Are you getting me now? And the axon snaps it on the spiral ganglion. So when it snaps it on the spiral ganglion, what happens? Afferent fibers fuses together to form the cochlear nerve I gave you. From the cochlear nerve, what happens? They snaps on the ventral and dorsal cochlear nucleus. From there now, from bilateral cochlear nucleus, from there they form what we call a trapezoid body by dissociation in the pons. So when they dissociate in other level, are you getting now, they snap on the what superior olivary nucleus. So when they dissociate and snap on the superior olivary nucleus, I've said it before, but same side from the ventral and dorsal cochlear nucleus can snap on the same side of the superior olivary nucleus. So a contralateral fiber and same side fiber. From the superior olivary nucleus, fibers from here goes and snap on what we call the nucleus of lateral lemniscus. From the nucleus of lateral lemniscus, it now goes through the lateral lemniscus fiber down to the what inferior colliculus, the nucleus of the inferior colliculus. From here also fibers, which is the inferior branchium, also carries fiber from the inferior nucleus of inferior colliculus to medial geniculate body, and from medial geniculate body to the what cortex, area 41, 42, area 22. So I hope it makes sense to you and you understand it. If you are new to my channel, make sure to subscribe, turn on your post notification button so that anytime I drop any video, you'll be sure to be notified. So I'll be seeing my next video. Keep watching me all the time. This is where science lives. This is where I explain everything to you and you understand. Bye bye.